The MIRaGe project is about trying to understand how Plasmodium falciparum, the malaria parasite, survive during the dry season. Because we know that during the dry season there is very little transmission, so the only place where the parasite can be is within human beings, typically in asymptomatic infections, and it can survive for months in these infections. It's still a very puzzling question as to why, or how, I should say, how do parasites survive during the dry season? Because we know that, or we suspect there is very little transmission during the dry season, so the only place where the malaria parasite can be is within these asymptomatic infections. So somehow the parasites survive for at least 8 months in those asymptomatic infections during the dry season. Well, basically we ask the question, who transmits malaria? These asymptomatic people during the dry season, are they able to actually transmit the parasite onto someone else? And do they do it better or less well during the dry season compared to the wet season? So we want to know these asymptomatic infections, how many gametocytes are there? And are these gametocytes effective? Can they produce a oocyst in a mosquito? Can they be transmitted? So in practice, we have started a longitudinal study in a village in the URR. And during the wet season, we get a blood sample from everybody every 6 weeks. During the dry season, we actually started a cohort on about 60 people. And we collect blood samples from these 60 people monthly over the entirety of the dry season. So that overall, we cover the entire year in that particular village.
Is to ask you for a little bit of blood, and we will put in this tube up to here, up to the first line here.
And with these blood samples, we do various experiments, including membrane feeding assays that determine whether the parasite will be transmissible from these blood samples. We also collect the infected red blood cells for DNA work, including whole genome sequencing, and RNA work for transcriptomics. And finally, the plasma is kept as well, basically to analyze the immune response. And this will allow us to basically analyze the host pathogen interaction all year round, wet season versus dry season, and try to see how the parasite can adapt from one season to the other.